My name is Dr. Claire Golléty. Welcome to the seminar on measuring benthic primary productivity and carbon flow in intertidal habitats. First, I'll discuss primary productivity and carbon flow, giving a few definitions and why should we measure carbon flow. Then I'll discuss different methods to use to measure primary productivity and carbon flow, ending up with incubations using benthic chambers. And finally, I'll discuss a few questions that can be addressed using this method. So what is primary production? It's the production of organic matter by autotrophic organisms. A commonly made mistake is to confuse it with biomass, which is the size of the compartment doing the production. And so we're interested in what is called productivity, the amount being produced during a time interval, and in this particular case for photosynthetic organisms. I'm assuming you're all familiar with this particular relationship, but the reason I'm showing it is because I want to define clearly what is gross primary productivity, which is the net production of organic matter going this way, and respiration, the reverse process which results in the production of ATP. The difference between these two processes is net primary productivity. Why should we measure primary productivity? First, to understand biodiversity ecosystem functioning relationships. And for example, who are the major primary producers or the sources of carbon flow in a particular system? Using this example from a rocky shore, you have here 3 different compartments, and the arrows tell you who is the major primary producers in this particular system, the fucoids. It can also help understand issues dealing between species diversity and functional group. For example, in this particular example, do we mean all fucoids that can be found in this type of systems, or only individual systems will be responsible for, individual species, will be responsible for this type of flow? It also helps understand what is the basis of food chains and how sustain different food chains depending on the quality and or the quantity. Expanding on this rocky shore example, if you compare a sheltered versus an exposed site, you see that the same compartments are the bases of these two food chains, but the primary producers are responsible for different carbon flows in either case. So that's what we're interested in, the magnitude and who is responsible for that magnitude. So how do we measure this? What we're interested is this particular amount, but it's very difficult to measure, accurately at least. So instead, it's much easier to measure chlorophyll A, the amount of chlorophyll A activated, or at least a proxy for it sometimes, and even better, oxygen and CO2 fluxes. So different methods that have been used to measure primary production in coastal habitats. A first one that is used, even though it's not reliable, is the harvesting method, which consists in harvesting at 2 points in time and calculating the change in matter, either fresh or dry way. This is commonly done for macroalgae and salt marsh plants. In this particular case, I've illustrated with macroalgae. This, at time 1, you harvest, for example, this much, and at time 2, you let it grow back and harvest again, and your difference in weight is supposed to be your production. Now, there are several problems with this approach. First is you're actually measuring an increase in biomass, not a production. Your sampling actually modifies the system under study, and at best, with many precautions, you're only measuring net primary productivity. A second method that is carried out in vitro is measuring carbon fixation using a carbon-14 method. So it's done using incubations under light and darkness, like any method that involves incubations. And you're measuring carbon fixation from a label substratum, usually bicarbonate. There are still problems with this method. Your organisms are isolated from the habitat, which means you are not taking into account potential interactions with the environment or other organisms. For macroalgae, you can often just use a piece of thallus, which again can have issues when upscaling to a whole individual or even more to a community. And there are issues with the initial hypothesis, especially for intertidal species, assuming that bicarbonate is a substratum use. It could be that they are capable of using CO2 in the air, for example. I'm going to illustrate a bit more the issue of using this method on Microphytobenthos. Even though you could use it on the whole community, Microphytobenthos or biofilm is composed of many organisms attached or associated with the substratum by a matrix made of mucopolysaccharide. And by taking it in vitro, you lose this matrix that has an important role in the interactions of the organisms within that community. So you lose this aspect. Another method that is also in vitro measures oxygen production, again using incubations. This time you measure oxygen fluxes using a probe. Still a few problems with this method. Your organisms are still isolated from the habitat. For macroalgae or seagrasses, again, you only use a piece of thallus or at best a small individual. And this is illustrated here in this small incubating chamber where a piece of macroalgae has been placed in it. And for Microphytobenthos, it is, again, a bit better in that you can bring cores back to the lab, thus making measurements on the whole community, but you still are constrained by a few conditions, such as measuring under constant temperature. Another issue is that you have to convert your oxygen production to carbon production. This is done using photosynthetic quotients that are based on the literature, and little knowledge is available on how they are valid from one organism to the other. An improvement is to use indirect in situ measurements using fluorescence. This measures photosynthetic efficiency and potentially relative electron transport rate depending on the conditions under which you do your measurements. This is done, for example, using a diving PAM. For macrophytes, again, you can only measure that on pieces of thalli. You're interested then in local processes, such as is there any photoinhibition undergone by the organism under study or recovery from a stress. For example, this is what has been done here on an individual of the macroalga Ascophyllum nodosum. The blue areas show high tide, whereas this is low tide. The blue line shows the amount of light, and the red squares are the photosynthetic efficiency measured at different points in time, which show how the effect of photoinhibition affects the individual, and then there is immediate recovery as soon as water comes back. For Microphytobenthos, you do have access to the whole community, and you can then understand processes like photosynthetic efficiency or migration. You still have a few problems with this method. It is difficult to compare between organisms, again, because of comparing results from a community to thallus pieces. It can be tricky to convert the results to carbon production, although it has been done on Microphytobenthos here by coupling the mean relative electron transport rate measurements to carbon fluxes measured directly, and this graph shows the variability that exists between these two measurements. Finally, a more direct approach is direct in situ measurements, where you can use first incubations in water, either by using rock pools as your incubating medium, or benthic chambers. This is a benthic chamber developed by a team in New Zealand, where in the intertidal they can install their chamber at low tide and then carry out their measurement throughout the high tide. In both cases, you measure either oxygen fluxes in the water using an oxygen probe or taking water samples, or you can measure carbon dioxide fixation, again taking water samples from which you measure pH and total alkalinity. Either cases, you do an incubation under light, which is net primary productivity, and then under darkness you get respiration. There are a few constraints with this method compared to the previous ones. You still have to convert oxygen production to carbon production if you measure oxygen. You can be faced with intensive laboratory processing following field sampling, especially if you want to measure pH and total alkalinity. And it might involve intense field work or even diving, which might not be available to all teams. Then you can also use incubations in the air this time. The metabolism measurement is then measured as CO2 fluxes at the air substratum interface, and the CO2 concentration is measured using a CO2 infrared gas analyzer. Under light, again, under a clear dome, you measure net primary productivity. Under darkness, using an opaque dome, you measure community respiration. And the slope is your CO2 flux, which is very convenient. And then the sum of these two processes, again, gives you gross primary productivity. Again, fewer constraints compared to the previous methods. You do have some weather restrictions, since the circuit used cannot be exposed to rain, and you do need a certain amount of light to carry out your measurement. As you can see in those pictures, you do have to find ways of transporting your equipment depending on where you're doing your measurements, and it can lead to potentially long field work hours depending on the system you're working on. Benefits from using incubating chambers. You are performing in situ studies. You measure direct carbon fixation, either CO2 in air or pH and total alkalinity in water. You can perform comparisons between different intertidal communities, and you are measuring at the community level for any system. And you can perform direct measures of ecosystem functioning that can then be coupled with biodiversity measures. And you can even perform manipulations of your system within your chamber, and thus improve your understanding of the system. So, because of all these benefits, the chambers have been used to address many questions, from testing the effects of different environmental factors to establishing carbon budgets, but also to understand the contribution of different compartments to carbon fluxes, and to understand relationships between biodiversity and ecosystem functioning. This has been done on different temperate communities, sediment microphytobenthos, intertidal rocky reefs dominated by microalgae, seagrasses, and salt marsh plants. So let's first look at the effect of environmental factors, and starting with the effect of light on gross primary productivity. And let me first walk you through this model of how photosynthesis or gross primary productivity varies as a function of light or irradiance. And first start with a linear relationship that then reaches a plateau, which is defined as the maximum photosynthetic rate. These two parameters are then used to define E or IK, the light saturation coefficient. And at the individual level, too much light usually results in a decrease in photosynthesis, which is called photoinhibition. So this can be measured by performing chamber measurements either during sunrise or sundown, so that you get access to natural changes in light intensity, or using natural filters of light, like I did during my PhD. This is the chamber viewed with a filter on top of it. And this has been done using this frame that allows maintaining here different filters, so that different intensities of light can be controlled. And this is the type of result we produced with it. Here shows gross primary productivity as a function of light on the measurements done on macroalgae. And it shows that first at the community level, there is no photoinhibition. Similar measurements have been done previously on microphytobenthos on a muddy sand estuary. So you can see that the same type of relationship was observed. Again, no photoinhibition, and with the added value here that by performing the same type of measurement at different times throughout the year, they looked at how the photosynthetic parameters varied with the season. You can also look at how photosynthetic parameters vary, whether you're at low tide or high tide, which is what had been done here on the seagrass community. Then you can look at the effect of air temperature on gross primary productivity and community respiration, this time performing measurements under saturating irradiance, so that light is not a factor taking place. This done on macroalgae again, gross primary productivity at the top, community respiration at the bottom, and the fluxes have been plotted as a function of the average air temperature measured on the day when the fluxes were measured. And in both cases, this exponential relationship can be established, similar to what has been shown on the microphytobenthic communities or a seagrass community. You can also look at how the temporal variations that take place during a tidal cycle. First, across the immersion period, here look on a macroalgae dominated reef again, and this shows how the effect of time since the beginning of immersion results in two different relationships when you look at gross primary productivity on the left or community respiration on the right, or how the change from immersion to immersion affects the carbon fluxes, here illustrated on the seagrass community. So on the bottom graph you have water height in black with the low tide marked by the gray section and the white rectangles showing light. And at the top you have carbon fluxes, with white being gross primary productivity and black being community respiration. And you can notice that even though you have more light at low tide, you have much lower fluxes in the air in this particular system than in the water, similar for respiration. You can also look at seasonal variations of carbon fluxes. So first, on macroalgae-dominated communities, gross primary productivity at the top, community respiration at the bottom, in both cases as a function of days of the year. And in both cases, you see that they follow a nice sinusoidal relationship. And then this can be compared to measurements done before on muddy microphytobenthic community. Again, gross primary productivity at the top, community respiration at the bottom, same type of relationship. You can observe that the magnitude of the flux is quite different between microphytobenthos and macroalgae. So you can see you have a direct comparison between the two habitats. What is similar is when the maxima occur. For gross primary productivity, it takes place around June-July when the light maximum is reached. Whereas for respiration, it is shifted towards July-August when the temperature maximum is reached. You can also look at the effect of a gradient of grain size on gross primary productivity. This is a small estuary that has the following grade cycling from sand to mud. And measurements of the effect of temperature on gross primary productivity and season were performed at 3 stations along this gradient, showing that the going from mud to sand actually dampens the relationship that is usually observed. Using all these measurements to study different environmental factors, you can then put together carbon budget. So I'll illustrate what has been done on a microphytobenthic community. You have here, and the negative values, benthic community respiration, on the positive values, gross primary productivity, in black under optimal irradiance, that is without taking the effect of any cloud cover, and in gray under measured irradiances. Going back to this sediment grain size, you can establish the same budget at each station, and you see that again you have a damping effect as you go towards the sandy station. And by taking the difference between gross primary productivity and respiration, you get net primary productivity for each day of the year. And you then observe that at the sandy station you have a slightly autotrophic system with 6 grams of carbon per meter square per year being produced. At the intermediate station, you have again an autotrophic system, whereas at the muddy station, you have a heterotrophic situation where there's a net consumption of -9 grams of carbon per meter square per year. Something similar has been done on an escophyllum nodosum dominated community, and you get these fluxes for gross primary productivity, community respiration, and net primary productivity, again under measured irradiance. This shows again the much higher magnitude of fluxes compared to the microphytobenthos, but it has the advantage because you can manipulate the system and especially perform measurements with or without the canopy in the chamber. You can then understand what is the role of this canopy in the budget, which is quite high. You can also understand how the fluxes vary between high and low tide, with most of the gross primary productivity taking place at high tide, but most of the community respiration taking place at low tide. And this is further illustrated here with this graph of net primary productivity in grams of carbon per meter square per day as a function of day of the year, which shows that at high tide the budget is largely autotrophic, so a net production of carbon, whereas at low tide the budget is heterotrophic, resulting in a consumption of carbon. So with this last example, I've hinted at the fact that you can understand better the contribution of the different compartments to the community carbon flow. Going back to the example of the effect of grain size, these authors wanted to understand why there were two such different situations, and they suspected it was due to the role of the bacterial community. So the community at this place is split into the bacteria, the meiofauna, and the macrofauna, all contributed to the benthic secondary production. And by coupling the chamber measurements to bacterial production measurements in the lab and establishing biomass values for microphytobenthos, the bacteria, the meiofauna, and the macrofauna, they were able to establish that the bacterial production was responsible for up to 69% of the benthic secondary production, and that the contribution of the bacteria to the secondary production varied with grain size. More importantly, it was the benthic community respiration that was mostly due to the bacterial respiration. So at the sandy station, there was low organic matter, resulting mostly in the production of new bacterial cells, whereas at the muddy site, there was an excess of organic matter, resulting in high bacterial respiration. In estuaries, where you have important developments of macroalgal strands, you might be interested in understanding how these macroalgae contribute to your flat carbon budget. This was done by these authors by performing measurements with and without the macroalgae, which showed that these macroalgae can be responsible for 3 to 10 times the gross primary productivity of the microphytobenthos and 3 to 20 times the benthic community respiration of the microphytobenthos. Now this is at hourly rates. What happens to the entire annual budget? When looking at the gross primary productivity, it showed that so with black being the measurements without macroalgae and gray with the macroalgae, it showed that the macroalgae were responsible for 24% of the annual gross primary productivity budget. But when looking at the net primary productivity, it showed that the macroalgae were only responsible for 12% of the annual budget, showing that actually, on an annual basis, the role of the macroalgae is limited, and this was due to their role being restricted mostly to the summer period. You can also look at the role of macroalgal canopies. During my PhD, I established that Ascophyllum nodosum is responsible for 76 to 100% of the community gross primary productivity and 79 to 98% of the community respiration. And by performing similar measurements not only on an Ascophyllum nodosum stand, but on Fucus vesiculosus of the same tidal level and Fucus serratus just underneath, it showed that in each case, the canopy dominated both the primary productivity and the respiration of each zone. Now, this is quite significant and could only be explained by understanding the role of the microscopic epiphytes in these macroalgae-dominated canopies. The canopy, in all cases, were responsible for very high respiration, resulting in up to 65 to 80% of the carbon produced that was respired instantaneously. This is quite significant. So I'll illustrate this. At low tide, when we performed our measurements, under light, we measured high gross primary productivity with a relatively high respiration, thus ending with a positive, relatively small net primary productivity. Under darkness, there is no gross primary productivity, but a high respiration resulting in a high negative net primary productivity. So over a 12-hour cycle at low tide, you're likely to have a negative net primary productivity. If we assume at high tide that pretty much the same thing happens, except you probably have a higher gross primary productivity. So you still have a positive but not very large net primary productivity under light, but then this time under darkness, again no gross primary productivity, high respiration resulting in very high negative net primary productivity. And this again results in a 12-hour cycle at high tide of a negative or maybe null net primary productivity. So you end up with an annual cycle of negative net primary productivity, which is in complete contradiction with the high biomasses that are being accumulated in those sites. So clearly there's something wrong with this picture, and what we think it's that we're missing the role of these microscopic epiphytes linked to the production of high extracellular polymeric substances by the macroalgae during low tide. These are produced as a mechanism against desiccation and end up being a substrate for high metabolic activity by the microscopic heterotrophs living on the surface of these algae, thus explaining the high respiration that we measure at low tide. So this still holds true, but at high tide it means things are different. And instead, when the algae do not need to produce such amounts of EPS, the substrate available to the heterotrophic organisms is reduced, so that respiration is much lower, thus ending under light in a much more important and positive net primary productivity, and under darkness, negative but much smaller negative net primary productivity. So you finally have a positive net primary productivity underwater, and thus an annual carbon budget that makes sense, and does end up in the accumulation and exportation of organic matter. So with this, using this in situ method, we were able to improve our understanding of the system. It was assumed before that 10% of the net primary productivity of the system is consumed locally, and then 90% is exported. We think things are slightly different. You have to consider gross primary productivity, where 70% of it on average is respired directly, either by the algae itself or its microscopic epiphytes. 10% is still consumed on the spot, but there's only then 20% left to be exported to other systems. The last type of question that can be addressed is to improve our understanding on the relationships between diversity and ecosystem functioning. This was done, for example, by when looking at the effect of canopy loss on biodiversity and ecosystem functioning recovery. On an Ascophyllum nodosum stand, we had 4 areas: a control area that remained intact throughout the study, an area where Ascophyllum nodosum was cut at 80 centimeters from the holdfast, one where it was cut at 20 centimeters from the holdfast, and one where it was removed and later a fucus vesiculosus canopy developed instead. I won't go into more detail in the study, but basically we coupled the metabolism measurements to length of Ascophyllum and diversity measurements to test the effect of harvesting Ascophyllum on the habitat structure provisioning, the carbon fluxes, and the benthic biodiversity, looking at macrofauna and macroalgae. And the results briefly were that the 20 centimeter harvesting and the canopy removal had very similar and significant effect on all three types of parameters. The biodiversity recovered after a year and a half, independently of which of the two canopies were present, whether it was Ascophyllum or fucus. In terms of habitat structure provisioning, there is still no recovery today after 7 years after the treatment took place, and this could have consequences for the pelagic diversity, such as fish that might use those areas for refuge at high tide. And the carbon fluxes were the variable that actually recovered fastest, since after 6 months, once the fucus vesiculosus was established on the no canopy area, showed no difference at all with the other areas, thus showing the functional redundancy that exists between these two types of canopies. Another question that has been tackled using these chambers is to test the effect of multiple stressors on diversity and multiple ecosystem functions. This was done by performing the same experiment at different areas throughout Europe. So at the red spots, but obviously on land, not in the sea. At each area, we had a cross-factorial design to test the effect of both canopy presence, absence, and or disturbance or no disturbance. And on each quadrat, by using this form of the chamber that had a square base, we could have on the same quadrat the carbon fluxes coupled to the actual diversity measurements. Again, I'll skip directly to the main results, which were that there was a worse effect of the disturbance when the canopy was absent only for the gross primary productivity, not for the community structure, the net primary productivity, or the benthic community respiration, which showed the importance of measuring multiple ecosystem functions, which was made easy using this method. The work I'm currently carrying out here in St. Andrews is to look at the effect of salt marsh restoration in the estuary ecosystem functioning. So this is the Eden Estuary north of St. Andrews, located here. And it used to be covered in salt marsh, but now on the south shore it's restricted to this small area. And it is now used as a donor for transplanting salt marsh plants at these locations. And so we know that in terms of habitat structure, the restoration is successful, but we had limited understanding of how the biodiversity and the ecosystem functioning might be restoring. So for that purpose, I carried out metabolism measurements at different sites along this coast. And briefly, although this is still ongoing and in the process of being analyzed, it does seem that both for gross primary productivity here and benthic community respiration here, there is, if you compare what is measured on the natural site, this is bare mud flat, and then going up years since the site was transplanted, there seems to be a return to functionality. Again, this has to be analyzed further, but it's a first look at the results. The last study I'll illustrate is linking biodiversity and ecosystem services at different spatial scales, and this is done in the CBEST project. And this time, the benthic chamber measurements have been coupled to eddy covariance tower, which also measures CO2 fluxes, but at over a much higher spatial scale. This is one of the sites where it is carried out. So on the soil marsh areas, each point represents a quadrat where the benthic chambers were deployed. The cross shows where the eddy covariance tower is, and the red area is the footprint over which it is measuring CO2 fluxes. And the analysis will consist in comparing whether using the measurements from the small scale area with the chambers and the larger scale area with the eddy covariance tower results in the same magnitude of fluxes and the same understanding, or whether the two the upscaling might not be linear, basically. And that's it for now. So thank you very much for your attention, and I'm happy to be contacted if you have any question on what I've presented or further questions regarding the chamber. Thank you.